everyone. The management and the Department of Plant Biology and Biotechnology, Loyola College, warmly welcomes you to the 4-day international webinar series on infectious diseases, biology, diagnosis, preventive strategies of coronaviruses. This webinar is very much the need of the hour, and the department is happy to conduct this webinar series. Let us begin with a prayer. Loving Heavenly Father, we thank you for your protection and providence at this unprecedented time. We implore your grace and mercy at this time and pray in a very special way for our fellow beings. We also give this webinar into your hands. Bless it and prosper it. In your most precious name we pray. Amen. I now invite the head of the department, Dr. Ravindran, to welcome the participants.
Most distinguished resource persons, my dear faculty members and participants, good morning and warm greetings from the Department of Plant Biology and Biotechnology, Loyola College, Chennai. It is my privilege and honor to welcome you all to this international webinar series on infectious disease, Arboviruses, biology, diagnosis, therapy. I wholeheartedly welcome all the resource persons, Dr. Saravanan Thangamani, Professor, Center for Environmental Health and Medicine, Preventive Biology Lab, SUNY, Upstate Medical University, USA, Dr. Prof, Principal Scientist from USA, Dr. Raghavan Palaniappan, Senior Scientist, USA, Dr. Choravat Ranganathan, Ahmedabad, India, and my special word of welcome to Dr. Saravanan Thangamani, today's resource person. I am extremely happy to welcome all the other participants and my dear faculty members. Loyola College is very much grateful to you, all the resource persons, for accepting our invitation to deliver a lecture through this webinar. I also welcome all my colleagues on this occasion. It is my special duty to welcome all the participants. Really, we are more than...
So, mute it, Dr. Saravanan? Mute करता है. Dr. Saravanan? Yeah. Now it's correct.
All right.
Okay.
Okay, I'm not sure what happened, but
Yeah.
I'm going to share my screen now.
Okay, you can begin. You can start.
Right. All right, good morning, everyone. Sorry for the glitch there. And I would like to thank the organizers, specifically Dr. Agastin, for giving me the opportunity to talk to you all about infectious disease threats and a little bit of highlight, a little bit about what I'm doing in my laboratory. So I want to start with a disclaimer that the opinions that I present here today are of my own, and it doesn't represent my employer and also my funders. So whatever I say, it comes from my mouth and I'm responsible for that. So I have divided my presentation into these multiple different chapters, I would call it. I would give a brief overview about emerging zoonotic diseases, and I will follow with the zoonotic disease arbovirus origin, and then I'll go on to talk about future arboviral threats. That will be the half of the presentation. The rest half of the presentation, I would like to kind of give a highlight about what I am doing in the laboratory, particularly on the zoonotic diseases. And then I will finish up with some concluding thoughts of what I think we should be doing considering whatever is happening at this time, particularly with regard to COVID-19. So let me start with some definitions to start with. So what does it mean, a zoonotic disease? Zoonotic diseases are infectious diseases caused by a virus, a bacteria, and parasites that spread between animals to humans. 6 out of every 10 known infectious diseases are actually spread from animals to humans, actually. And 3 out of every 4 new or emerging infectious diseases in people come from animals. So I think that's what we call zoonotic diseases. It's like a spread between animals to people and vice versa. But how do we get this zoonotic disease? There are different ways we can get the zoonotic disease. One thing is from the direct contact, coming in contact with the saliva, blood, urine, mucus, fecus, or other body fluids from an infected animal. Example includes petting or touching animals, and bites or scratches. And we could be indirectly exposed to a zoonotic disease by coming in contact with areas where animals live and roam, or objects or surfaces that have been contaminated with germs. Example include aquarium tank water, pet habitats, chicken coops, barns, plants and soil, as well as food and water dishes. The third most important one I would consider is that the vector-borne disease transmission. It's primarily if you get bitten by a mosquito or a flea or a tick, you will get transmitted by a disease. You will get an agent that will cause disease. And foodborne as well. Eating or drinking something unsafe, such as unpasteurized milk, undercooked meat or eggs, or raw fruits or vegetables that may be contaminated with feces of an infected animal. You can get it sick as well. And drink or coming in contact with water that has been contaminated with feces from an infected animal. So these are the primary ways how a human could actually acquire a zoonotic disease. So when I say zoonotic disease, I told that it's actually transmitted from an animal to a human, right? So that's what the animals are considered as reservoir host. So what does it mean, reservoir host? Those are animals that become infected with virus and keeps the virus circulating in its bloodstream for as long as possible without dying from an infection. So those are called reservoir animals. So what are the primary reservoir animals that we currently are aware of that causes zoonotic disease? Bats are the primary ones that causes Ebola disease, Ebola virus, SARS, of course, SARS-CoV-2 as well, MERS, rabies, Nipah virus, and Hendra virus. And birds, they do transmit bird flu. And rodents, primarily virus pulmonary syndrome, plague, rat bite fever. Dogs, we all know it transmits rabies to humans. And, ticks causes Lyme disease, Powassan virus, Crimean Congo hemorrhagic fever, tick-borne encephalitis. And mosquitoes, we all know it transmits malaria, dengue, West Nile virus, Zika, Chikungunya. So these are called reservoir animals. So how do we define the scale of disease spread? This is the last, a series of definition slides, and that will be over. So what do you call an outbreak? An outbreak is when an illness happens in unexpected high numbers. It may stay in one area or extended more widely. So this is an example. The map actually shows an example of how SARS-CoV-2 kind of exploded into a pandemic. It started as a small outbreak in the city of Wuhan. And then it kind of spread to other provinces. That's what we call an epidemic. So epidemic is an outbreak over a larger geographic area. A sudden increase in the number of cases of disease in a particular geographic area beyond the number health officials typically expect. And that's what happened. So initially it started as an outbreak, and then it actually expanded to different provinces throughout the China. And then pandemic is a disease outbreak that spreads across countries or continents. It affects more people and takes more lives than an epidemic. And pandemic is the highest level of global health emergency that signifies widespread outbreaks affecting multiple regions of the world. So these definitions, I think the SARS-CoV-2 kind of fits all these definitions. It started as a small outbreak, then became an epidemic, and then within a month or two, it became a pandemic. Now the entire world is suffering from COVID-2. So with that, I would like to actually highlight a couple of zoonotic diseases, just to kind of emphasize how human and animal interface creates this confluence of events in such a way that we get terrible diseases. So obviously, I want to start with SARS-CoV-2 because everybody is aware of it. And SARS-CoV-2 was primarily, is a coronavirus, is primarily known to be circulating animal to animal. In this case, it's bat to bat. And there was a spillover. It is expected, or it's believed that there is a crossover transmission from bats to wild animals like possums or pangolins that live in wet markets in China or Wuhan province. And what happened was that there was a spillover transmission from animals to humans. So initially, the transmission was between animal to animal transmission. So humans were not involved in the picture, but then animal to human transmission happened. That's what we call a zoonotic transmission or a spillover event. A spillover event is an event where a disease or agent is actually jumping from one species to another species, called a spillover event. So when animal to human happened in Wuhan, that's the smaller outbreak happened. And then as soon as that outbreak happened, human to human transmission occurred in different ways, through community contact, through mass gatherings, through travel. And when a sick patient go to the hospital, the healthcare workers that treat them got infected. That's called nosocomial transmission or hospital-acquired infection. And the same thing is with a sick patient, transferring the virus or provide a healthy patient, or well, I wouldn't call a healthy patient, non-infected patient gets the COVID from a sick patient. So these are multiple ways how this SARS-CoV-2 kind of spread from bats to wild animals to humans, from a single human to multiple different humans and throughout the world. So SARS-CoV-2 is a perfect example of a pandemic, or I would call it a zoonotic disease. It kind of exemplifies every single epidemiological definition. So next big thing that I want to kind of talk is because it kind of helps to explain this, differential event that happens is Ebola. Everybody knows that it's a dangerous disease to get. It's a hemorrhagic fever, 50% case fatality rate. It was originally discovered in 1976 when 2 consecutive outbreaks of fatal hemorrhagic fever occurred in different parts of Central Africa. The first outbreak occurred in the Democratic Republic of Congo, formerly called Zaire, in the village near the river Ebola, and that's how the name came as Ebola virus. The second outbreak happened in Sudan, and depending on the locality, the case fatality rate can go anywhere from 50 to 70%. So how does it happen? Similar to SARS-CoV, animal-to-animal transmission happens between bats to bats, bats to monkeys, or bats to smaller mammals. And that is a spillover event that might happen where a human get exposed to it. Human try to hunt the monkeys or try to poach them or try to get their dead ones for hunting or other purposes. The human get exposed to it. And then once the spillover event happens, it becomes a human-to-human transmission. Like SARS-CoV, again, here the transmission is different. It is traditionally because of unprotected contact with the blood and body fluids, and an unprotected healthcare worker, and the traditional funeral practice. These 3 are the major drivers of Ebola spread in West Africa and Central Africa. And as I said before, the first outbreak happened in the Democratic Republic of Congo. That's earlier it was called Zaire. So this is where the first Ebola was isolated, and that's why it's called Zaire Ebola virus. The red circles represent Zaire Ebola virus, and the blue ones represent the Sudan virus. So these 2 were the first 2 viruses that were Ebola viruses that were isolated. And as you can see here, Zaire Ebola virus pretty much dominates the entire West Africa and Central African countries. There are 2 other Ebola viruses that cause significant human disease. That's called Thai forest Ebola virus and also the Bundibagyo Ebola virus, but majority of the Ebola cases are Zaire Ebola cases. So the clinical presentation is like fever, severe headache, muscle and joint pain, and severe diarrhea and vomiting will happen, and then unexplained hemorrhaging, bleeding, and bruising from every single orifice of a human body will happen. And then the case fatality rate, as I said earlier, is 50 to 70% rate. And the map here, if I can take your attention to the map down here, is the dotted lines represents the geographic distribution of the fruit bats that are compatible or that are capable of transmitting or harboring this Ebola virus. And you can see that it actually goes all the way pretty much sub-Saharan Africa, South Africa, and then to Asia, Southeast Asia, and the northern parts of Australia as well, and to the Pacific Islands. So it covers a wide geographic area. And the colors that are highlighted in orange, those are the countries, so those are the geographic areas that have been documented to have Ebola outbreaks. And the countries that are represented in light orange, where that means that in those countries, humans were found to contain or found to have antibodies for Ebola virus, which means that they are seropositive, which means that they have been exposed to Ebola at some time during their life. You can imagine that most of the Southeast Asia, Indonesia, and Philippines, they do have zero evidence that they probably had Ebola of some sort there. And the ones that are in the blue ones that are like you see in the United States, those are travel cases or Western Ebola case. Western Ebola is an asymptomatic. It is not known to cause human disease, and it's actually primarily originated from Philippines, but ended up in the United States. And the monkeys that came from Philippines ended up in a facility called Reston facility near Virginia, Washington, D.C. That's how the name came by. I don't know if you have read the book, The Hot Zone, that documents how this Reston Ebola case was actually discovered. And that's the reason why it's called Reston. It's actually an asymptomatic infection, and it is not known to cause human disease. The other one I would like to share is the Nipah virus disease. I think most of you would be very familiar with this disease because there was a small outbreak in Kerala, India in 2018, where 15 humans were, succumbed to the disease. Again, how does the transmission happen? Bats are the primary reservoir here again. So it's bat-to-bat transmission happens initially, and then humans get exposed to this Nipah virus if they actually take the contaminated sap or fruit. It's in villages, it's very common where they actually take palm sap to actually consider as a quote unquote alcoholic drink. When they try to drink this contaminated sap that may contain the saliva of the bat, then the human gets the virus as well. Plus, bats, when the pigs or the swine population in pig farm, when the pigs eat the contaminated fruits and contaminated material, they acquire the Nipah virus as well. So initially, when the first case was actually identified in Malaysia in 1998, where primarily the pig farmers were the ones that get sick. And that's how they found that actually it is pigs are also a reservoir. And then eventually they found that the bats are a potential reservoir to transmit the disease. And it's a relatively new disease. It causes encephalitis, and it can cause about, the illness presents for 3 to 14 days with fever and headache, disorientation and mental confusion. And the symptoms can progress to coma within the 24 to 48 hours. Again, the case fatality rate here is 40% to 75%, but this rate is depending on the local capabilities for epidemiological surveillance and clinical management. My understanding is that when there was an outbreak in Kerala, I think they did a great job in containing the virus. That's why the outbreak was very limited to 15 deaths. But there have been sporadic outbreaks in West Bengal and Silgura, India in 2001, where 66 cases were identified of that 45 deaths. That's pretty high case fatality rate. And oftentimes in Bangladesh, because I think they all share this, Brahmaputra Delta, they do have this similar disease happening there. So it is kind of restricted in a geographical area within the Southeast Asia, primarily India, Malaysia, and Singapore, but this is a dangerous disease to get. So what are the primary factors influencing the disease emergence? Obviously, you talk to any different scientist, they would come up with their own laundry list of factors that actually cause this emergence of zoonotic diseases. But if you ask me, this is what I would actually come up with that. And we can go with that. Population growth and urbanization, in my opinion, is a big thing that influences the how we are actually bringing close to the let's say forest. We are population is growing tremendously, so we need more space to construct houses. So what do? We cut the forest, we cut all the trees, and then we start to live in the forest, which means that we are putting us so close to the wildlife, and that means that whatever the disease the wildlife is having, we will be getting as well. So population growth, urbanization is always a bigger problem. And then obviously these are connected to economic development and land use, breakdown in public health systems. Needless to say, SARS became a bigger problem. SARS-CoV-2 became a bigger problem because of breakdown in public health systems. So I don't think I have to explain too much on that. The other thing, again, I can use SARS-CoV as an example, is the human susceptibility to infection, which means that if you are immunocompromised, you are susceptible to pretty much 99% of the diseases. If you have diabetes, obviously you are susceptible to so many different diseases. If you have hypertension, so there's a lot of conditions that actually make you sick to a particular disease. Also, lack of public health services. So this connects to the breakdown. So breakdown means that you have public health services, but nobody is implementing it correctly. The lack of public means there is no public health systems there. If there is an outbreak, there is no system to even take care of it. And international trade and travel is a primary thing that is a causative agent that you can tell that nowadays, you can go from Australia to all the way to California within 24 hours, which means that if someone is sick with SARS-CoV-2, they take a flight all the way to California, which means that they can start another cycle in California within 24 hours. So that's the power of the travel. Well, unfortunately, it's a negative power, but that's the power of the travel in disseminating disease. And also the change in climate and ecosystem. Climate change and the weather change is a big driver of zoonotic diseases. Primarily, it drives away the reservoir host, that has the viruses in them, to come close to the human habitation because their forest is being destroyed. So we are coming closer to their contact. Also, the change in weather, change in climate, change in pattern within a year, it allows the mosquitoes and ticks to actually behave differently in such a way that we will get longer exposure to mosquitoes. The mosquito season will be very long because of the pattern changes, the weather pattern changes. And the microbial adaptation is change. I think I have an example to tell in the couple of slides to come by, is that viruses, they continuously mutate themselves in such a way that our body can't recognize that, I'm trying to, the way it is that they are evolutionarily adapted to change themselves in such a way that they can survive longer in nature. So the microbial adaptation is a bigger problem when we try to design, let's say, antibiotics or vaccines or diagnostics. It's a big problem why we can't actually have vaccines for a particular virus very easily, is that because they keep on mutating. And then if we develop a vaccine against a particular virus A, within a year, there will be A1 will be there, which means that the vaccine will not work against the A1. So that's the biggest problem. Also, the geographic expansion of the reservoir. So as I saw earlier, the reservoir are those animals that actually has the capability to harbor the virus or any agent without succumbing to the disease. So it can transmit it. So the geographic expansion, we all know that mosquitoes keep on expanding to new areas. And it's the same with the animals as well. They keep on moving, they keep on migrating. As humans migrate, animals migrate, wildlife migrates as well. So with that, I want to segue into the zoonotic disease arbovirus origin. So what does it mean, arboviruses? Arboviruses are arthropod-borne viruses, which means they're viruses that are transmitted by arthropods/insects. Not all arthropods are insects, though, I have to warn, but for the sake of convenience, I can tell that. So the arbovirus come in different shapes, different infectivity rates. Also, the arthropods, even the grasshoppers can actually transmit viruses to plants. So they don't we are not talking arbovirus means it doesn't only transmit disease to animals, it actually can transmit disease to plants as well. So majority of the arboviruses, 99.99% of arboviruses are RNA viruses. They belong to the family Togaviridae, Rhabdoviridae, Rheoviridae, Flaviviridae. So an example of Togaviridae is a chikungunya virus. I'm pretty certain everybody in India is familiar with chikungunya virus. It belongs to the family of Togaviridae. And I'm pretty certain everybody is familiar with the Zika virus or dengue virus or Japanese encephalitis. Those are flaviviruses. So these two, Togaviridae and Flaviviridae, are the major disease-causing families. And there is one virus out of thousands of arboviruses. There's only one DNA virus, which is called African swine fever virus. It primarily, as the name suggests, it primarily targets swine population. But this is a highly economically devastating virus, which means that if a particular pig gets infected with this virus, they really have to cull the entire farm because it can easily spread to the next pig very easily, which means that the pig farmers will lose a lot of money on having this virus. It's primarily in the Northern Africa and Southern Europe, and I think now it is slowly appearing in China as well, but this is the only DNA virus we know of arbovirus in the arbovirus world. So again, as I said, Togaviridae, Flaviviridae, Buniviridae are the major arbovirus family. And as you can see here, Togaviridae is a little bit of, it's about 70 nanometers in size. Flavivirus can range anywhere from 40 to 60. And Buniviridae are a little larger viruses. It's 80 to 100 nanometers. And Togaviridae, it's a positive sensed single-stranded RNA virus. Flavivirus is almost identical. It's a positive single-strand RNA. However, the Bunyaviruses are negative single-strand RNA viruses. And obviously, Togaviridae, there are about 29 viruses, and Flavivirus is about 68, and Bunyavirus, there are about 318. Of these, majority of the human disease-causing viruses, they actually belong to Flaviviridae. So what are the primary vectors of arbovirus transmission? Pretty certain everybody knows this is a mosquito, and this is a very obscure looking arthropod called ticks. So I kind of put the difference between these two are the primary transmitters of arboviruses. They pretty much transmit. Sandflies do transmit some viruses, but compared to mosquitoes and ticks, they don't cause too many diseases. So mosquitoes are capillary fielders, as the image shows that it actually put this mouth part, or we call it proboscis. It's like a syringe. It goes deep into the skin, pierces through the skin, and goes to the capillaries and puncture the capillaries and try to sip the blood from the punctured capillaries. However, on the other hand, here the ticks, they are like, we call it pool feeders. So they put their mouth part deep into the subcutaneous skin and then try to lyse the skin in such a way that it recruits all the blood to the mouth part area and then siphon it like it has a straw, and then it is sipping, let's say, a soft drink. So that's why also mosquitoes only feed for 5-10 minutes. On the other hand, ticks can stay attached to a human or a mammal for 4 to 5 days. So that's why I call mosquitoes are fast food, and then ticks are like gourmet food. They are very patient to attach to a human, and they take their blood for 4 to 15 days. So some of the major mosquito-borne viruses that we all know that cause significant human disease. Everybody knows dengue virus, or dengue fever virus. It's present throughout the world. South America, Central America, North America, Asia, Australia. And the zoonotic vector or zoonotic reservoir is a non-human primate in the forest cycle. And the transmission, the primary mosquito that transmits are Aedes aegypti and Aedes albopictus. Those are present in India. And it affects nearly 390 million infections per year. So you can imagine that out of that, 30 to 50% are symptomatic. So it is a severe debilitating disease one could have. The next one, everybody's memory is Zika virus. Zika virus belongs to the antigenic group, Spontavini group of viruses. Again, it is present throughout the world. wherever dengue is present, it's also present because it is transmitted by the same mosquito that transmits dengue. In addition to that, Zika virus could be sexually transmitted vertically from mother to fetus. Okay, vertical transmission mother to fetus. Thousands to millions, depending on the year, and primarily it started appearing in the radar since 2013. So the cases are now exploding. So I think that it will be, probably it will run very close to the dengue viral cases. The other important virus is the West Nile virus. It is present mostly in North America, Middle East, Africa, Europe, and Australia. The zoonotic reservoir here are birds, because these Culex mosquitoes are ornithophilic mosquitoes, which means that they like to feed on birds. Humans are accidental hosts. So they like to feed on hosts when they don't find birds, and if a human is around that area, it will feed on humans. So there are about 10,000 cases per year, but in America, this is a big problem we are having. And Japanese encephalitis, it is prevalent in Asia and Australia. And birds and pigs are the reservoir here. They are the ones that actually they perpetually carry the virus. And it's transmitted by Culex mosquitoes. Again, they're ornithophilic mosquitoes. They prefer to feed on birds. There are about 70,000 cases per year. It's a highly dangerous disease to get because it causes meningitis and encephalitis. It has even if you survive this disease, you will have long-term neurological sequelae. So that's your life will be really, really in a terrible condition if you get Japanese encephalitis. And yellow fever is primarily in Africa and South America, and non-human primates are the reservoir host. It's transmitted primarily by Aedes aegypti, and there are about 130 severe cases per year. But the good thing about the yellow fever is there is a vaccine available. You can have vaccine. If you go to any travel clinic, I believe you'll be able to get a vaccine against yellow fever. So I know that most of this disease that I actually showed here are transmitted by Aedes aegypti and Culex mosquitoes. These are the two mosquito species or genus that actually transmit the vast majority of mosquito-borne diseases in the world. And you can see here, whatever you see in yellow, blue, and red, those are the places where the Aedes mosquitoes thrive, which means that they can, in those areas, they are capable of transmitting Zika virus, dengue virus, yellow fever, chikungunya, and a lot of other bacterial and protozoal diseases as well. And you can see in India, pretty much everything is there, but for the area of Rajasthan, closer to Pakistan, I think we see green, which means that they are not suitable for, they don't have suitable growth conditions for these mosquitoes. That's the only area in India we don't have Aedes aegypti and Aedes albopictus mosquitoes. And when you move on to the Culex species, as you can see from the map, pretty much almost 80% or 90% of the globe is actually are suitable for Culex pipiens, Culex confaciatus, and Culex tarsalis mosquitoes. And obviously India, pretty much a majority of the India has Culex confaciatus, and closer to the North India, probably about Delhi, all the way to Kashmir, you have Culex pipiens mosquitoes. So these mosquitoes can actually create a major problem to India, particularly you have both Aedes aegypti and albopictus, and then you have the entire country has Culex mosquitoes. It is a perfect combination. I would call it a confluence of events in such a way that it can create a havoc, particularly from the mosquito-borne disease perspective. And I'm not talking about malaria here, that's a bigger problem, but I'm focusing on viruses, but they can actually cause significant disease to humans in India. So I want to kind of give a brief introduction to chikungunya virus. It's one of my favorite viruses, I can call it, because I made my career out of that. When I became an independent faculty for the first time, that was a virus that I chose to work with it. And I was lucky to have great mentors who actually trained me with that. And also I was at the right time. I became an independent faculty when chikungunya virus was really at the peak. So I consider that it was one of my favorite viruses to work with. So it's an alpha virus. As I said, it's actually a tocoviridae. It belongs to tocoviridae family. It's an enveloped virus. It is a positive sense single-strand RNA virus. It's about 11.5 kilobase. Viruses are not really, they don't have long genetic material. It's very small. It's easy to sequence. It's easy to do a lot of work with them. And their structure, schematically, they look like this. So they do have 4 non-structural proteins and also 5 structural proteins. It's very easy to work with it, and it's very easy to make chimeric viruses. Chimeric viruses are more like a infectious material, but they are not live virus. It's basically you create a plasmid, you put all the promoters there in such a way that they become actually, they replicate, without considering them. We don't consider the live virus, but they are chimeric viruses. They are like pseudoviruses, but they still work the same way as the regular virus. So chikungunya, the term actually came from Tanzanian Makonde dialect, which is called disease that bends up the joints. So when someone gets a chikungunya virus, you can tell that they always bend because they have back pain, severe debilitating back pain they will be experiencing. It was first isolated in 1953 from the blood of several febrile patients and several mosquito species, including Aedes aegypti in Tanzania. And you can see that chikungunya presents, in a clinical setting, it presents multifaceted conditions, like it causes joint pain, fever, headache, myalgia, cutaneous manifestation, joint swelling, digestive symptoms. But as you would see that 92% of the human cases present with joint pain. And this is not just an acute infection. In some cases, in majority of the cases, it's a chronic infection, which means that you will have initial chronic pain, and you might think that it kind of went away from your body. No, it actually never goes away. It goes to your kneecap. It goes into the synovial fluid. And synovial fluid is actually an immunologically privileged environment in such a way that the immune cells, they don't go into the synovial fluid to attack any foreign body. So that's where the virus goes in, hides there. And when someone's immune system gets weakened, when they get older, this flares up again. So in my opinion, I consider chikungunya as a chronic disease. It can attack you anytime when someone's immune system kind of wanes down, it comes back and attacks it. And this also, chikungunya is very interesting, the fact that it attacks the knee joints, it attacks all joints and go to the synovial fluid and create all the havoc. And also it attacks the muscle, and it actually damages the muscle cell as well. And that's one of those things that it actually attacks the muscle and also the joints. So it's a perfect combination of events not to have in a long term. So how are they transmitting in nature? So in nature, chikungunya, we call it sylvatic cycle, is what is happening in the forest. Nature, the chikungunya virus circulates between non-human primates, or monkeys, and arboreal mosquitoes. Because these monkeys, they tend to live on the top layer of the canopies of the trees. So there are certain mosquitoes that live only on the canopies, and they essentially feed on these monkeys and then circulate the virus. And some of these viruses are Aedes africanus, Aedes furcifer, Dalizae, and Vittatus. So they kind of circulate in nature. So this is the animal-to-animal transmission. So between monkey to mosquito, and then the cycle goes on like that. But humans become an accidental exposure. Like we are so interested in, okay, I want to go to Africa. I want to go and see African safari. I want to go into the woods to do all these, wonderful things, trekking. So what happens is that when human population goes into the deep of the woods, they get exposed to these mosquitoes accidentally. They get bitten by them. Then what happens is that they finish their vacation in the woods and they go back to their country or go back to wherever they came from. And now they have virus in their body. And then they have this competent mosquito, Aedes aegypti and Aedes albopictus, in that environment, which means that now the humans are a reservoir. These mosquitoes can pick up this virus from the human and then transmit to another human. So you start in another endemic cycle there. So that's called urban transmission, where the mosquito transmits the virus between humans to mosquito. In sylvatic transmission, it's wild animals, primarily monkeys, non-human primates to mosquito. That's a sylvatic transmission. So this cycle sometimes gets spilled over because we are trying. That's what I call that human behavior. Anthropological behavior affects the way how the zoonotic diseases are actually happening. Technically, the sylvatic transmission happens by itself, the urban transmission happens by itself, but then because we are trying to, from urban, we are trying to go into the rural areas, and that becomes the spillover event happens. So when I mentioned about the mutation, how viruses are mutating in such a way that, they add more problems to humans, this chikungunya is again a very good example. Also, I call chikungunya, it's a great example of a globalization of a viral event or viral epidemic event of an epidemic potential. So what happened was that until, I can go back 2 slides. So until 2004, the chikungunya essentially circulated in East Africa, Southeast Asia. It never left. It was sporadic outbreaks here and there. I'm pretty certain nobody ever heard this virus before 2014. But what happened between 2004 and 2005 is that a mutation event happened in this virus in such a way that alanine at 226 position became valine. So this one change in the E protein actually made this particular virus to infect Aedes albopictus mosquito and then cause an epidemic potential. So prior to that mutation event, only one mosquito transmitted this virus. But after the event happened, both the mosquitoes actually transmit the disease. So I have to explain you here. That's why I put this small mosquito picture here. So what happens when a chikungunya virus, let's say I'm infected with chikungunya virus, a mosquito feeds on me. So it takes the virus through the proboscis all the way to the gut. It goes to the midgut. So virus has to replicate in the gut. It has to infect the epithelial cells in the gut and then has to escape the gut, go to the hemocoel. Hemocoels are, it is like blood because they are invertebrates. They do not have a regular blood like we have, but they do have open circulatory system. So the virus gets into the hemocoel, goes all the way to the salivary gland. And then in salivary gland, it's a complex architectural. It replicates there and it has to go to the salivary duct and then it is ready to be dumped again into a human. But it has to cross so many different cells and organs to be able to go to the salivary gland and then transmit again to the human. That's what we call barriers of infection. So when a chikungunya virus gets into the mosquito, gut is a barrier. It has to escape the gut. So those are the gut barrier. And then it has to infect the salivary gland, that have an infection barrier. And it has to escape the salivary gland to the human. So for a virus to go through all these things, it really has to, And not like mosquitoes don't transmit Ebola. Mosquitoes don't transmit SARS. Mosquitoes don't transmit any other viruses that, not known to be transmitted. The reason is that they have a strong immune system in such a way that they don't allow all the viruses to be multiplying in the body and then go to the salivary gland. It only allows certain viruses to go through it. But this particular mutation happened in such a way that a new mosquito became a competent vector, and then it created a big problem throughout the world. So what happened was that prior to that mutation event, this virus was circulating primarily in the East Coast, Eastern African coast, and in India, of course, Pakistan, and Southeast Asia. But now this is the global map of what we have. You can imagine that pretty much the entire world is, I would say that they have chikungunya. The one that you see in the blue color are travel-associated cases. We do not have local transmission. However, you would see in the United States, in the Florida, this is the Florida Panhandle, that we have found mosquitoes that actually carry virus and also locally transmit the virus. So it's a perfect example of a globalization. So if I can summarize that, it's illustrated very clearly here is that prior to 2004, the virus was circulating primarily in Africa and Southeast Asia. But that mutation even happened. It exploded. Primarily, it happened in the La Reunion Islands. La Reunion Island is in the Indian Ocean. It's a tropical island where Europeans, they like to do their vacation. They want to escape their cold weather to come to tropical weather. And they all got exposed to this particular mosquito that had this mutated virus. They went back to Europe and they started a cycle there. And then everybody went back. So that kind of started a cycle. And right now, we have 3 different lineages of this virus circulating. It's a perfect example of how a virus can mutate itself and become a global event. So with that example, I want to kind of segue into tick-borne viruses with the time we have. Ticks transmit greatest variety of viruses. However, not all of them actually make humans sick. But there are a good number of them that actually make people really, really sick. One being Crimean Congo hemorrhagic fever. It is primarily in Asia and Africa and parts of Europe.
And I think in the state of Gujarat in India, there are cases of Crimean Congo as well. And 10 years back, a new virus was discovered in China, which has a case fatality rate of 30%. And in America, 6 years back, they discovered another virus called the Heartland virus. It's primarily endemic to America. And Kayasunu forest virus. It's a flavivirus. As the name suggests, it's actually a Kayasunu forest in the state of Karnataka, if I'm right. Actually, I have a couple of slides for that. And Alkurma is a dangerous virus, and the Powassan virus is dangerous, tick-borne encephalitis. So most of these tick-borne viruses, they have a high case fatality rate. So it's pretty interesting or pretty important to actually understand those viruses as good as we know about the mosquito-borne viruses. So I want to kind of give a kind of very quick snapshot of what is happening. Is that Crimean Congo hemorrhagic cases were first discovered in 1944 in the Crimean Peninsula. That's how it is. And this map, you can tell clearly it kind of exploded to entire continent of Africa, southern Europe, and some parts of India as well. And the way how it transmits in nature is, again, it's a tick that transmits called Hyeloma marginata ticks. And they like to feed on small mammals, and they like to feed on wild domesticated animals as well. On top of wild animals, they like to feed on domesticated, and that's how humans get infected with this tick, because they like to particularly the farm workers, when they come close to the domesticated animal, goat or sheep or cow, the ticks will actually feed on them and will cause them the disease. So again, there are two stages of the disease. One is a pre-hemorrhagic phase, the other one is a hemorrhagic phase. Pre-hemorrhagic phase is like any other febrile illness, but if you're lucky, you will survive the pre-hemorrhagic phase. But if you're not lucky, you will go to the hemorrhagic phase, where you will have petechiae, hematomas, or hemorrhage from nose, gastrointestinal, and urogenital. So it's as good as, I would say, an Ebola case. So the case fatality can be anywhere around 50 to 70% as well here. So I want to give a brief introduction about this Kayasunu virus. It is a neglected virus, in my opinion, because we don't know much about this virus. It was originally discovered in 1957 in the Kayasunu forest in the state of Karnataka, if I'm right. Yes, that's Karnataka. Yeah. So it was discovered by three scientists called Telford Work, Rodriguez, and Praveen Bhatt. So at that time, they were at the National Institute of Virology, and they discovered this virus. And Telford Work used to work for the National Institute of Virology at the time, and he's the guy here, and this is Praveen Bhatt. He then became the CDC. He then became the director of CDC later. So again, it's a tick-transmitted disease. How do humans get in contact with that? Is that monkeys and small mammals are the reservoir here. So when a human gets into contact with the dead monkeys, or probably hunting them, they will get exposed to the virus. Also, these viruses, these ticks actually feed on domesticated cows and cows as well. The people who go to the woods or the forest to collect firewood, or the forest ranges, those are the people who are at risk. So this is the grim picture that I want to present here, is that it was discovered in 1957 to 59, and then initially there were about 56 cases every 2-3 years. And then after 1966, there are no data clearly for me to go look into that. So what I found is that from 2014, there is a data that I can track onto that. And you can clearly see that in 2014, there were like 166 cases of that 3 were fatal. But in 2020, well, let me put 2019, there are 445 cases, of that 15 is fatal. In 2020, up to April 24, there's already 234 Kayasunu forest disease cases, of that 5 are fatal. So this disease is on the rise in India, in the South India primarily, where these ticks are highly, highly prevalent. So I think this is something that everyone should be watch out for, particularly if anyone is interested in doing this kind of work. I think this is right down the alley. So future global threats, in the interest of time, I'm going to skip a little bit fast here, is there are multiple diseases that people can predict will become a major outbreak, but in my opinion, I consider Usutu virus could be a major virus to be that we should be worried for. It actually belongs to Japanese encephalitis group of viruses, and it is primarily present in Africa and Europe at this time. Zoonotic reservoir is bird. Obviously, Culex pipiens are the vector the mosquito that transmit that. So it causes febrile syndrome, meningitis, encephalitis, and acute flaccid paralysis as well. Right now, number of human infections are not 100% clear how accurate they are, but we believe it runs from hundreds to thousands. And it shares so much, it's so much similarity to West Nile virus. I think that this Culex pipiens is present throughout the world. The Culex species are present throughout the world. So this has every single recipe that this could be the next big, I would say, like next big Zika virus. The other virus that everybody thinks could actually go big as well is Pondovini virus. As the name suggests, it's Pondovini group of viruses. It's present in Africa and North America, and it is transmitted by both Aedes and also Culex mosquitoes. So this is another potential recipe for disaster. And it is very close to Zika virus. It is also known to be sexually transmissible. So this is another thing that we are watching out for. It's in our radar. These two viruses, in my opinion, are in my radar, at least to kind of look for what is going to happen in the future. So with that, I'm going to skip directly to my research. I don't think I have enough time to go because I ran out of time, but I can give with this one slide what I'm really doing is that my lab does a lot of, I have a very large research portfolio. I do basic science, and then whatever I understand from the basic science, I try to translate that for vaccine development or therapeutics. And there is another arm that we are doing is the epidemiology and new virus discovery. So as far as the basic science is concerned, I understand the vector, host, and viral determines arbovirus transmission. And we are developing new animal models for emerging arboviruses because we need animal models to understand the pathogenesis of a particular virus. So we have to develop, each virus behaves differently. So we have to develop animal models for every single virus differently. And our lab is interested in, or we are actually identified candidates for transmission blocking vaccine in such a way that when a mosquito feeds on us, it will not be able to transmit the disease. So that's the idea of blocking the transmission of the virus. It's called transmission blocking vaccine. Also, we have developed, we have identified candidates that could be used for as a therapeutic option to arboviral infections. In the epidemiology, we do vector-borne discovery, vector-borne surveillance, and pathogen discovery. And we try to understand the impact of climate change on the geographic expansion of diseases in the state of New York. Recently, we started a citizen science program. We call it Got Ticks. If you are interested in knowing what we are doing, how we are doing, feel free to go. I'm an open book. I put all of my research on my website. You can follow there. If you're interested, you can contact me. So our research, lab research, is actually, it's very simple. So for a vector-borne disease to transmit, you need three components. You need a vector, and you need a pathogen, and you need a host. When all three come together, there's only one time they all come together, when a vector feeds on us. Because a vector needs to feed to transmit the disease. If it doesn't feed on you, it doesn't transmit the disease. So we are looking at that interface. We call it vector virus host interface. That's where the virus is being transmitted. So if we can understand what really happens in that one tiny little area where the mosquitoes are feeding, we can try to develop novel methods, novel countermeasures to stop the transmission. So that's the idea. So as it says, I work with mosquitoes and ticks, and I work with a bunch of viruses, including Chikungunya, Zika, Powassan, Tick-borne encephalitis, Heartland, and the Chinese Bunya virus. I'm going to skip a couple of slides in the interest of time. What I want to say is that okay, so this picture, I will finish it off here easily. So here, I showed you that these mosquitoes and ticks, they feed differently. One is a capillary feeder, the one is a pool feeder. So what my lab is doing is trying to understand this tiny little area. I call it foramen skin biopsy. What is happening? That's all I'm looking. And we've been looking for several years, but the reason is that skin is a physical barrier. Skin is the first organ the pathogen counters when a tick or mosquito feeds. Infectious agents are delivered there. And this is a nice picture, a schematic diagram of skin. You can see that skin has multiple different immune cells, or they're resident, they're already there. So the virus has to escape all of these immune cells in such a way that they can go to the lymphatic system. From the lymphatic system, they can go to the target organs. So how does the virus escape all these things? And how does the ticks or mosquito help the virus to escape this immune cell attack? And that's essentially what we are doing in the lab. So I want to kind of highlight what, one of the things that I'm really very proud of what we have done is we have reported for the first time microRNAs from an arthropod from the mosquito saliva. So we have identified 72 microRNAs in chikungunya infected mosquito saliva. Of that, 16 are very novel. And we have checked their capacity to neutralize the virus. Yes, we had shown it. Also, we have created genetically modified mosquitoes in such a way that could knocking out a particular microRNA, what would happen to its virus capacity to transmit the virus. So we are currently doing this study by generating genetically modified mosquitoes and by knocking out the microRNAs from them to understand. Hopefully we'll be able to develop therapeutic options based on the microRNAs. And for the tick side, basically what we are doing, as I said, we are looking at that 4 mm tick biopsy, exactly what is happening at the feeding site. We don't want to look any further. That's what we want to do. And then, as I said, the ticks secrete salivary proteins in such a way that facilitates the virus. So we are trying to identify which salivary factors facilitate the virus so that we can develop novel neutralizing factors for that. So I'm going to skip everything, and then one final step we have actually identified in the sake of time. So we have identified in such a way that the early immunological responses at the tick-powassan virus interface are pro-inflammatory with a marked increase in immune cell infiltrates at the tick feeding foci. And we have also demonstrated that the tick saliva facilitates virus transmission and dissemination. And we have identified microRNAs and mRNAs that are differentially expressed during the powassan infection. And we have demonstrated that IL-1 beta and IL-6 orchestrates inflammation at the time of virus delivery, which is then activated by TREM, goes all the way to the target organ. So we have done a lot of things, and I would appreciate if anyone is interested in what we are doing, please go ahead and read our publications. We'd be happy. And I would, in the next few minutes, I'll finish my concluding thoughts. And so if anything that the current COVID-19 has taught us, it is that we are not prepared for a pandemic event. And this is clearly evident. I wouldn't say that for any other country, but I can certainly say that United States is not prepared for that. And here are the diseases that WHO released a blueprint for priority diseases that they want everyone to develop vaccine. As you would see here, there are no vaccine available against these deadly diseases. We don't have for crime. We have an experimental vaccine for Ebola. Marburg, no. Lassa fever, no. MERS, no. SARS, no. SARS-CoV-2, no. Nipah, no. Rift Valley, no. Zika, no. So everything is no. So how are we going to improve our responses to infectious disease threats? We have to improve public health infrastructure, increase routine immunization, mass vaccination following a disease outbreak, and One Health approach to mitigate zoonotic diseases, surveillance of infectious diseases in human, animal, and also vector population, and building local capacity to diagnose and report cases of infectious diseases, and leveraging opportunities for informal surveillance using Google or Apple or ProMed or crowdsourced information to fed into the surveillance as well, and monitoring lab accidents and regular monitoring preparedness. Also investment in research and development for biomedical countermeasures such as vaccines, diagnostic antimicrobials, monoclonal antibodies, novel therapeutics, platform technologies. And also to strengthen the supply chain and improve systems for rapid distribution of countermeasures in the event of an emergency. Also, coordination between national and international efforts. These are very important. Again, these are challenges. These 12, I think people can add on more bullet points, but it's our challenges. But I consider these challenges are opportunities. There are tons of things we can do it. We don't know a lot of things. So for me, I kind of consider this an opportunity to kind of understand how to mitigate all these infectious disease problems. So with that, I would like to acknowledge my lab members primarily, because they made everything possible for me. Also generous funding, also my collaborators. And with that, I'll take any questions if you have. I'm sorry that I had to skip my research slides, but if anyone is interested, they can write to me or they can follow me. Thank you.
Thank you so much, sir, for that great presentation. So we have few questions here. Can I read it out?
Sure. Yes, go ahead. I will stop the presentation now.
So the first question is, why is that bats specifically are the initial hosts for major deadly diseases like COVID, Ebola, or Nipah?
Bats do have a very good immune system in such a way that it doesn't bother the viruses doesn't actually bother the bats. So that's a simple thing. Every animal has a different immune system. Also, the viruses have co-evolved with the reservoir in such a way that the viruses will not kill the host because it becomes a dead-end host. So it's a co-evolutionary pressure that the virus doesn't kill the bats. At the same time, the bats have a very good immune system that prevents the virus killing the bat. And that's another reason why it's so many different diseases as well.
So the second question is, why North Indian people have minimum COVID infection when compared to other parts of India?
Well, I have to tell that with COVID, every day is a new day. We learn new things every single day. So unfortunately, what we know today may not be correct tomorrow. So I think that it's not actually fair to compare North India versus South India in terms of the number of cases for COVID because it's too early to tell what is happening. So I would actually say that at this time, your guess is as good as my guess. I have no idea.
Thank you, sir. The third would be, what are the methods to control transmission of arbovirus?
So first method is actually preventing the reservoir, which is mosquitoes. So we have to, kill the mosquitoes. We have to eliminate the mosquitoes. Also, we have to develop vaccines against arboviruses. So those are the primary things we can actually control, how we can control arboviruses. Kill the mosquitoes, kill the viruses.
Thank you, sir. Fourth question, is there any antigenic variation seen in coronaviruses?
I would say yes. There's already multiple lineages that have been identified. I think there is also a talk that the lineage that we see in the New York came from Italy that is different than what is circulating in China. So certainly there is an antigenic variation that is appearing. As I said, that we are very early. I think probably if you ask me 6 months from now, I will have a very clear answer.
So the next would be, does the severity of viral infections has any variations with respect to ethnic or racial variation in a population?
Yes, I think this is something that I wanted to put as one of my emergent factor is that human demographic and racial disparities in sense that everyone's genetic makeup is different. No two humans are the same. It will be the same for racial profile as well, or not racial profile. It will be for independent race as well. So there is a discussion going on in the United States that African Americans are more susceptible to SARS-CoV than a Caucasian. So yes, it is highly possible. And I can tell that no two individuals are the same. So what is good, maybe, what is good may not work for other people as well.
Thank you so much, sir, for addressing all the questions here. We have few more questions, but I think we would...
I don't mind.
Okay, sir. I'll just... Sir, is it effective that... Okay, why mortality rate of COVID is more in USA than in other countries?
I would say breakdown in public health systems. Again, it's my personal opinion, but it's true. Yeah, I think it is to do with the breakdown in public health systems.
Yes. So there's a question, why most of the RNA viruses don't have vaccines? Because
they are highly mutated. They mutate very quickly. Like I said, the Chikungunya virus was a great example. All it needs is one mutation from alanine to valine, that actually made it a big pandemic event, or not a pandemic, a global event. So they mutate a lot, and because of the way the biology of the virus is such a way that they need RNA-dependent polymerases, and we all know that, like even a PCR reaction, it is prone to make mistakes. So that's how the mutation happens actually. So they change quite often.
Thank you, sir. The discovery of herbal therapy, is it possible to cure COVID-19?
I think it is possible. It is possible. I think that I believe that several scientists or several companies are looking into natural use of natural medicines to control coronaviruses.
Can we expect more re-emergence of zoonotic viruses?
Of course. I think that's something that I would like to emphasize, is that once this COVID is over, I'm pretty certain there will be another virus that will be waiting to infect us. So that's the nature of every few years, we always see something big happening. So in 2006, Chikungunya became a big thing. And I think 2008 to 9, we saw, I think Ebola happened Ebola happened, and then Zika happened, and then we have COVID. So it's like every few years, there is always an outbreak that happens that causes severe disease to humans. But I have to tell that we never learned from our past mistakes. We always check. So what I say is that we always do retrospective analysis rather than being proactive.
Thank you so much, sir, for that answers that you gave for all the questions. So I just leave few questions will be there, sir. We'll mail you and we'll get the answers from you, sir. So I'll hand over this to Agastin, sir, for vote thanks.
Okay, thank you.
Dr. Saravanan, I am really proud of you. India really proud of you for your wonderful job, wonderful research work you are carrying out in USA. And at this juncture, I would like to thank you for your an elaborative explanation where everyone can understand all your topic. And also, I thank you for your answering the questions raised by the audience. Yeah, and also, I thank you for your information on various opportunities and scopes of research. You mentioned definitely our audience will take up. Definitely will come with good results in the area of infectious diseases. And also, I thank everyone. Particularly, I thank Dr. Jacqueline Chinarani in the prayer, Dr. Ravindran in welcoming address, and also I thank the audience who put a lot of questions. I thank each and every audience. Okay, you made this as a possible work. Thank you very much. Thank you, Saravanan. Thank you very much.
Thank you. I appreciate listening.
Yeah, thank you. Now coming to the audience. Kindly fill up the feedback form and post it today itself before 6 o'clock. And also tomorrow, the same 10 o'clock, we'll start another interesting topic in the field of structural biology of coronaviruses. Okay? So thank you for being with us. Thank you so much. So with this, we come to an end. We close the webinar today. Thank you very much.
Sir, congratulations, sir.
Good night now.
Saravanan sir.